You're listening to CoachingCues podcast, where experts all around the world answer your most burning questions surrounding the wide topic of strength training. Every week we tackle the what, the why, and the how of one specific topic in just 15 minutes. Straight to the point, no fluff. So, without further ado, let's get straight into this week's episode.
My name is Adam Sharples. I am a Professor of Molecular Physiology and Epigenetics at the Norwegian School of Sports Sciences in Oslo, Norway. Our research lab has been investigating the question, does skeletal muscle have a memory? So, on today's podcast, I'm going to be talking about this specific question. Now, when you think of memory, you immediately think of the brain. Memories of past experiences sensed by the central nervous system and stored in the brain. So, for example, I would remember my son's or daughter's last birthday and what we did that day. When you add the word muscle to the term memory, however, you might also think about memory of movements in our bodies. Memories sensed by learnt movements like riding a bike, and that our muscles never really forget these movements. So, for example, once learnt, you would never forget how to ride a bike. However, we're going to talk today about a third type of memory, because I guess as coaches and athletes interested in training adaptation, we aren't necessarily talking about memories in the brain. We are interested in our muscle being one of the primary tissues in the body that can adapt to exercise training, such as increases in size after resistance exercise or improvements in metabolic capacity after endurance training. We also aren't talking about muscle memory of learnt movements. And actually, it's slightly unfortunate that the term muscle memory has been used to describe this type of skill acquisition, as this is really a motor learning or motor memory that is driven by the central nervous system, remembering the specific neuroactivation patterns that help acquire movement for a new skill, that once learned, like riding a bike, they're not forgotten. The memory we are talking about for this podcast, and for those interested in training adaptation, is where the muscle possesses a memory at the level of the muscle tissue itself. Our group has defined this type of muscle memory before as the capacity of skeletal muscle to respond differently to environmental stimuli in an adaptive or maladaptive manner if the stimuli have been previously encountered. This is an important definition because as athletes and coaches we are interested in if muscle tissue can remember previous exercise or training events and adapt more favourably in the future. For example, does our muscle remember a period of training when we're able to put on muscle mass, for example, or be in the best shape we've been in before? And how do we get that back if we've been injured or undertake a period of detraining, for example, during the off-season or even forced detraining due to the recent COVID pandemic, or actually even a period of voluntary abstinence from exercise? Then, is it possible that we can perhaps tweak our training to provide the longest lasting memory of an earlier exercise or training, so that when we do retrain after a period of detraining, we can adapt more quickly and return to full fitness? In other words, could we alter, for example, the exercise frequency, intensity or timing of training, leading to a memory of that prior exercise that lasts for longer? Hypothetically, addressing these important questions would allow the incorporation of this type of kind of optimized muscle memory training into an athlete's or recreationally active person's periodized training program. It could also help reduce the total volume or intensity of training required, meaning more time could be spent recovering, therefore perhaps reducing the risk of injury, or increase time spent on skill-specific tasks for a given event or sport. In simple terms, if you could train at a higher volume or intensity less frequently, and still have the same adaptation, this would be beneficial in freeing up more time for recovery or other types of training. So before we start to address these kind of applied implications of muscle memory, we have to look at the two main theories of how muscle memory has been proposed to work, theories that have both emerged from the scientific research within this field. The first theory is a so-called cellular muscle memory, and the second is an epigenetic muscle memory. And then there is also a degree of overlap between these two emerging theories. The first theory of a so-called cellular muscle memory came from work also from a research group in Norway at the University of Oslo. Now, before presenting this research, as a warning, this is where the podcast gets a little technical and some terms and jargon are introduced. And where this happens, I'll try and go back and ensure the meaning of these terms are also explained, but in the context of muscle memory and exercise. However, you'll have to bear with me at first. So, here goes. It was Professor Christian Gunderson's group at the University of Oslo that demonstrated in mice, treated with testosterone for 14 days, which led to an increase in muscle size, that they acquired new myonuclei in their muscle fibers. And these myonuclei were retained even when the mice stopped this testosterone treatment. Then when these mice subsequently underwent a mouse equivalent of exercise training in the lab, their muscle was able to hypertrophy, that is increase in size, more quickly than mice that had not previously encountered testosterone. What this tells us is that when muscle encounters anabolic or muscle building stimulus earlier, in this case by testosterone administration, even after a subsequent period of no anabolic encounter, i.e. the removal of testosterone, when the muscle exercises later, it can then adapt and get bigger more quickly. In this study, this effect was attributed to the finding that these muscle fibers retained these so-called myonuclei after the first earlier period of muscle growth or hypertrophy, and that then these myonuclei were retained when muscle returned back to normal size, so that when training occurred later, these nuclei helped the muscle grow more quickly. So what is it about these retained increases in myonuclei that help the muscle grow more quickly later? Well, before we kind of address that, it is important to understand what myonuclei are within our muscle and our fibers. As we are all probably aware, skeletal muscle contains muscle fibers, and these muscle fibers can be extremely long. So, for example, the sartorius muscle in the upper thigh can have fibers that are around 60 centimeters long, and it's estimated that these could have up to 40,000 nuclei. Nuclei are what contain our DNA, and an important site where our muscle fibers get the blueprint information to ultimately make new proteins for growth or to contribute to an improvement in function if we're talking about adaptation. So, for example, if our muscle needs new myosin, of course a crucial protein in the muscle that interacts with actin to produce muscle contraction, our nuclei contain several genes on our DNA sequence that code for different types of the myosin protein. For example, fast or slow myosin genes correspond to fast or slow muscle proteins that then produce fast or slow muscle twitch fibers. Then when our muscle needs new myosin, for example during an increase in muscle size after training, the muscle will communicate with the nuclei to increase the expression of the myosin genes that are then translated into increased protein by an organelle called the ribosome, which is outside the nuclei in the muscle fibers themselves. So the nuclei are key to control the process of gene expression, or kind of gene turning on, and then protein translation, which is the production of new proteins, that then ultimately affect how much protein we have in our muscle, that can in turn affect muscle size and its function. Now, the term myonuclei simply refers to nuclei within or inside the muscle fibers. And this is because muscle fibers are multinucleated. That is, as we have explained, they can contain many nuclei within each fiber. But this means muscle fibers cannot usually divide to create a brand new fiber that resembles the old one, and this is what's called post-mitotic. That is, the fibers cannot undergo mitosis or divide like single cells with only one nucleus could. Despite this, we know muscle fibers adapt to exercise, and therefore it really begs the question of how this is achieved without being able to add new fibers. And indeed, this is actually due to a resident stem cell that is located on the periphery of muscle fibers, and due to this satellite location, this cell is called a satellite cell. These cells can undergo cell division, therefore after a stimulus such as muscle damage from exercise, these satellite cells are activated, They divide and migrate to the site of that damage, and they fuse or merge to the existing muscle fibre and become new myonuclei. And satellite cells are the main source of new myonuclei in muscle after exercise. So now that we know what myonuclei are, you might say at this point, what is the relevance of these results demonstrating that muscle can retain myonuclei after testosterone administration, and not necessarily showing that this is the case after exercise? Well, as athletes and coaches, of course, we can't dope our athletes with testosterone. We're only interested in exercise. Well, to address the issue of the importance of exercise in stimulating retained myonuclei, other studies have also performed exercise in mice that include 8 weeks of progressive loaded ladder climbing that indeed evokes an increase in muscle size, followed by 20 weeks of detraining and another 8 weeks of later retraining. Unlike the testosterone studies, these studies with exercise demonstrated myonuclei were accrued, so increased with training, and persisted or retained throughout the detraining period. And then the other elephant in the room, I guess, is that the studies described so far were in mice and not in humans. So studies have more recently undertaken resistance training, detraining and retraining in humans and investigated the number of myonuclei in the muscle. And on the whole, these studies have concluded that myonuclei are not necessarily actually increased in the initial training period in humans, in one particular study actually, and therefore there was no retention of myonuclei into detraining like what was seen in the mouse studies. However, due to the nature of this type of extensive intervention study in humans that the authors can really be commended for, the study was in a small number of individuals who demonstrated quite a lot of variance in the way they adapted to the exercise training. So actually in a small subset of those individuals, they did increase their myonuclei number and retain them during detraining. But this effect was perhaps convoluted across the other participants who did not demonstrate this response. And this kind of varied response to the same type of exercise in humans is quite well documented. I'm sure you've perhaps heard the term responders and non-responders to exercise, and this is likely to have played a role in the varied responses observed in this particular study. Therefore, there is now a real need to conduct larger training, detraining and retraining studies in larger cohorts of humans. But indeed, if I was a betting man, I would perhaps say that in a larger group of responders to resistance exercise, that a cellular or myonuclear memory would be observed. But we really need more evidence to be able to confidently state this. And there is also quite a heated debate about whether satellite cells themselves are actually needed for hypertrophy, as we also see hypertrophy of muscle fibres when satellite cells are removed in their kind of mouse models of genetic ablations formed. However, it's not the purpose of this podcast to go into major detail, other than what is more clear is that for the maintenance of muscle and muscle function over longer periods of time, the muscle is reliant on satellite cells being activated and being incorporated into muscle fibres as myonuclei. So at this point we kind of now know what cellular muscle memory theory states and the research surrounding this. We will now move on to the second current working theory of muscle memory, and epigenetic muscle memory, which is the main area of my group's research at the Norwegian School of Sports Sciences. And to explain this, we first have to explain what epigenetics is. And epigenetics really refers to modifications to our DNA that are caused by encounters with the environment. Importantly, these modifications to our DNA can help the genes, and in our interest in the muscle, nuclei turn on and off. Earlier we discussed how nuclei containing our DNA and the information to turn genes on or off, known as gene expression, ultimately impacts the level and the function of the proteins that are produced from this DNA blueprint. Now, one of the major modifications to our DNA is called DNA methylation. Ourselves and others have demonstrated that endurance, resistance and sprint interval exercise can cause considerable alterations in this epigenetic modification of DNA methylation in skeletal muscle, They've also demonstrated that muscle cells in a culture dish can retain epigenetic modifications in the form of altered methylation to their DNA for a considerable amount of time after, for example, encountering earlier inflammation. Therefore, as a group, we kind of came up with a hypothesis that retained epigenetic modifications might also be involved in muscle memory in humans after exercise within the muscle. Therefore, as mentioned, if these DNA modifications can help genes in our muscle nuclei turn on or off if they are retained after exercise, it might mean our genes can still be turned on or off long after we have exercised. So with this in mind, we undertook 7 weeks of resistance exercise training, 7 weeks of detraining and 7 weeks of retraining in human participants. These participants were able to increase their lean muscle mass in the lower limbs by about 6 to 7% during the first training period, and then lost their muscle back to pre-exercise levels during the detraining period. And then they gained around 13% of an increase in lean muscle mass during the later retraining period, which was about a 50% larger increase in lean muscle after this retraining period compared to the earlier training period. In this study, we analysed over 850,000 sites on the human DNA isolated from the participants' skeletal muscle for the levels of DNA methylation, and discovered distinct patterns in how the genes on our DNA were chemically marked or unmarked by this methylation modification during these periods of training, detraining, and then later retraining. Most interestingly, one of the groups of the genes lost these methylated tags, became untagged, during the muscle growth following exercise training, kept them off or retained this untagged profile after a period of no exercise and detraining, and then lost even more methylated tags during the second retraining period. This untagging of DNA is typically associated with switching genes on or increasing gene expression. So we also observed that the genes with this retained untagging then also demonstrated enhanced or even greater gene turn-on profiles after the later retraining compared with the earlier training period, suggesting that the DNA in the muscle remembered the previous phases of training and muscle growth. Furthermore, some of the genes with these retained epigenetic profiles even remained activated through periods of muscle loss during detraining, again suggesting that muscle remembered the previous phases of training-induced growth. And what's more, this memory meant these genes were further enhanced or activated in later retraining and associated with the largest increase in muscle size. Some of the genes we identified to have these epigenetic memory signatures had not been linked to muscle growth before, and as a consequence of this study, we've now more recently looked at some of these genes in more detail, and confirmed that they are also important in muscle growth. Overall, these studies from us suggest that epigenetic signatures via DNA methylation are retained after earlier training into the detraining period, and then can influence how genes responsible for muscle growth are turned on during later retraining. And we are kind of still unsure whether this type of epigenetic retention occurs with training, detraining, and retraining from endurance exercise, because of course these studies to date were in resistance exercise. However, the likelihood that this will occur, I think, is perhaps probable, as we also know that aerobic exercise or endurance training evokes this kind of untagging effect of DNA methylation. However, whether these are retained during detraining and retraining require future investigation to confirm this. And I guess also the DNA modifications from endurance or aerobic exercise are likely to be on different genes than those modified with resistance exercise. For example, genes that control metabolism rather than genes we identified to be involved in epigenetic memory of resistance exercise that are kind of genes responsible for growth or tissue remodeling. However, future research using endurance exercise as a mode of training during these training, detraining, and retraining interventions are really crucial in the future to confirm these hypotheses. One other caveat to this research is that there are several other epigenetic modifications that can occur in muscle after exercise, And these can also affect how easily our muscle is able to access the DNA to turn our genes on or off in response to retraining if training has been encountered before. These include histone modifications, and histones are the proteins that help us fold DNA into its organized structure within the nucleus, and also help unwind the DNA when genes need to be turned on or off. Therefore, these modifications do also require future investigation and are really likely to be key other epigenetic players in epigenetic muscle memory. So finally, I guess I mentioned earlier that the two theories of cellular muscle memory via retention of myonuclei and epigenetic memory via retention of DNA methylation would also like to converge and influence each other, especially given that epigenetic modifications such as DNA methylation could occur in either myonuclei or also the nuclei in important regenerative cells like the satellite cell as mentioned earlier. And actually, work from a group in the US from Kevin Murak has started to look at DNA methylation profiles after muscle growth in rodents in different nuclei populations after training and detraining in an attempt to investigate the convergence of these two theories of muscle memory. They've also further confirmed the studies we have performed in humans demonstrating that there is a retention of DNA methylation in rodents into the detraining period after an earlier training period. But interestingly, they also find that the methylation of myonuclei demonstrate different epigenetic retention profiles to all the other types of nuclei located in the muscle from different cell types like satellite cells or fibroblasts, which are cells located in our extracellular matrix, kind of those collagen networks that help us transmit force produced by the muscle to our tendons. So further work really now needs to be conducted on the epigenetic profiles, especially in those important muscle stem cells, the satellite cells, as these cells are likely to play a key role in the retention of epigenetic information in our muscle, given that they are the main source of new myonuclei in our muscle in response to different types of stimuli. So, given all this information, we are kind of now hopefully at the stage where we are broadly understanding the two main theories of muscle memory at least, and we can now bring it back around to our kind of initial applied implications of muscle memory. And actually perhaps on a bit of a disappointing note, because at the moment, to go from what we know about the mechanisms of muscle memory and how we could harness this knowledge into an applied setting, for example, creating a longer lasting memory of the muscle so that we can adapt more quickly when we train in the future, requires a lot more research to make this leap. And we talked earlier about if we could perhaps alter the exercise frequency, intensity or timing of training that would lead to a memory of exercise that lasts longer. So is there a specific protocol we could perform or our athletes could perform that would mean more nuclei are accrued for longer, or the epigenetic modifications are also retained for longer, so that when we undertake this training again we can respond or adapt more quickly or even maintain this adaptation for longer? Then if you could train at higher volume or intensity less frequently and still have the same adaptation, this would perhaps be beneficial in freeing up more time for recovery. But I guess really there's much more research that needs to be done on varying the type, timing and intensity of exercise and which of these stimuli evokes the longest retention of our myonuclei or retention of epigenetic modifications, so that when retraining occurs later, that this leads to the most enhanced gene turn-on profiles and therefore adaptation or further greater adaptation in terms of muscle function. I guess overall, the good news is that muscle memory seems to be real, and in the next few years, as researchers, will try and harness the knowledge of muscle memory we've gleaned so far to improve our performance in athletes. So hopefully you can watch this space.
Thank you for listening to CoachingCues podcast. If you would like your question to be answered by an expert, please head to coachingcues.org/ask. See you next week.